Okay, well, very warm welcome to everyone today from the various places that they're coming from. My name is Chris Thorne and I'm the Senior Manager for Field Marketing here at Twist. So before we begin, just a little bit of housekeeping. As you've probably noticed, you're all muted and the lines will stay muted during the webinar. If you do have any questions, please do submit them via the Q&A box at the bottom, and then we'll take some time at the end of the webinar with Dr. Crosetto to answer those questions along with his team. Following the presentation, and once we've completed the Q&A, if you have the time, you'll be redirected to a brief survey and be very grateful for any feedback that you can offer in that. Okay, so it gives me great pleasure to introduce Dr. Nicola Crosetto from the Science for Life Laboratory and Karolinska Institute in Stockholm, Sweden. His primary research interest is in understanding the origin and mechanisms of formation of genomic alterations in cancer cells, focusing on the interplay between genome organization, transcription, and genome fragility. Over the years, he's been an active developer of molecular methods now widely used, including several methods for highly resolved visualization of DNA and RNA molecules in cells and tissues, including the method that we're going to be discussing today, IFISH. And so on that, it gives me great pleasure to hand over to Dr. Nicola Crosetto.
Thank you very much, Chris. Excited to be here. Welcome, everybody. So today we are gonna talk about how to make fish probes using Oligo Pools. And in the backstage with me is Eleni Galali and Gabriele Girelli, that were really the leading forces in developing the IFISH pipeline that I will be presenting to you. And today's webinar is gonna be structured in two parts. First, I'll give you an introduction to the IFISH pipeline that I just mentioned. And in the second part, we'll have a sort of more hands-on, virtual hands-on guidance to how to design probes using a freely available web tool that Gabriele developed in the lab. But before we further proceed, I just like to briefly break ice by asking you to quickly answer these questions so that I can have a sense of the audience. Here it is. Great. Nice. Good, good. The newcomers are winning. Very good. Okay. Good. Everybody casted their ball. Great. So pretty much the majority is new to fish. So I'll try to give a little bit of background and try to proceed by assuming that you don't know the details. So that's great for me to know. All right. So let me introduce IFISH to you. But before I do that, I'd like to briefly give you a timeline of the techniques we are talking about. So I guess most of you are aware that in situ hybridization and particularly fluorescence in situ hybridization techniques have been out in the field for now quite a long time. But it is actually only in the past decade that there has been a major step forward in the field thanks to actually the development of technologies to produce high quality oligonucleotides such as the one that Twist uses. And the use of oligos and particularly Oligo Pools in the context of fish technologies was actually pioneered by the Tim Wu lab at Harvard. And in 2012, they introduced the oligo paints that then were later also used as a basis for the high throughput single molecule fish technique called MERFISH, developed by the Wang lab at Harvard as well. And over the years, we have been also pretty much active in this arena, and we have developed this IFISH pipeline that I'll be presenting to you really as with the goal of making Oligo Pools and the various tools available to a broad community of people. But before we go into the details of how it works, I'd just like to show you in this slide what we think are really key advantages of oligos and oligo probes compared to the standard in the field. And particularly, if we talk about FISH, we talk about mainly DNA FISH applications, particularly in diagnostics, where conventionally people use probes consisting of double-stranded DNA fragments cloned in bacterial artificial chromosomes. Now, oligo probes have several advantages over these sort of standard technologies, in the sense that they can be both used for DNA and RNA FISH. They are single-stranded. This provides a huge benefit, particularly for DNA FISH, by increasing the signal-to-noise due to easier annealing. Most importantly, they are faster to make, easier to make. And if you're interested in narrow genomic regions to visualize, definitely they are much better to be much better choice. And last but not least, I would argue that the design and labeling of oligo-based probes is much more versatile, as I hope I'll convince you in today's presentation. So we developed, as I was mentioning, the IFISH pipeline, having really in mind the goal of providing a comprehensive set of experimental and computational tools to the community, all the way from designing the probes to performing the actual experiments. And so IFISH probes are suitable for both DNA and RNA FISH. We provide an easy probe design interface. You will see an example of how we can use it. We also provide databases of optimally designed oligos that we are continuously expanding. What is quite unique is that we made a repository of unique tested and validated, individually tested and validated probes. And also we provide a user-friendly protocols for both DNA and RNA FISH. And you can read more in our publications from last year. Now, so how does the IFISH works? I should first mention that the idea was built on how oligopaints and MERFISH probes were designed. But what we aimed at doing in our work was really creating a more versatile and universal platform that could bring you all the way from oligo pools, high quality oligo pools such as those produced by Twist, all the way to having ready to use probes. Now, these probes consist of pools of oligos that have the structure that I'm highlighting here with the mouse cursor. So they consist of a variable number of oligos. We typically use 96 for DNA FISH application, 48 for RNA FISH, but really there is no upper limit to the number of oligos you want to have in a probe. And each probe will consist of a target sequence that specifically binds to either its genomic target or transcriptomic target, flanked by 2 adapter sequences. So all the oligos in a probe will have a unique combination of these reverse and forward oligos. Now, when we then do a PCR step, and I'll tell you in a few seconds why we do that, we'll also extend the probe to have what we call a color barcode sequence here, where we can then anneal or dock a secondary oligo coupled with 1 or 2 fluorophores in order to detect the binding of the probe to its target. Now, if you have a complex oligo pool that contains thousands of these oligos with unique combinations of reverse and forward barcodes, what you can do is dilute the oligo pool, dispense it in 96 well plates, and then use PCR to really amplify only the oligos belonging to a single probe in each well. And ideally, you can do this for a P number of probes and make each probe in multiple colors see as you wish. How do you do that? By delivering essentially in each well a unique combination of forward and reverse primers that will extend the amplicon introducing the color barcode and the T7 promoter sequence. Then we use the T7 promoter to actually drive linear amplification of the PCR product into RNA. So this further amplifies the initial amount of material. And then once we have the DNA-RNA hybrid, and we hydrolyze the RNA strand, and so we end up having a fully single-stranded DNA probe that is ready to use, and it is exactly this configuration shown here. I want to remark that all the way from the splitting the oligo pool to the final probe, we are amplifying the material 9 orders of magnitude. So we start with femtomolar amount of oligos in the complex pool that you can purchase from Twist, down to a large amount of probes that can be used for multiple rounds and in different experiments. So that's really the power of this pipeline. So what can you use these probes for? We can visualize individual genomic targets as shown here. This is a large probe targeting 1 megabase region on chromosome 18 with 12,000 oligos in the probe, really a large probe comparable to commercial ones, giving bright signals. We can also target much smaller regions and visualize multiple loci simultaneously, as shown here. Each of these dots comes from a probe consisting of 96 oligos. And these signals are really almost close to the diffraction limit and are therefore allowing us to precisely localize the spots in the 3D space of the nucleus. We have also developed a new concept to visualize entire chromosomal territories by designing multiple probe sets all along the chromosome that then we pull together. And the result, as you can see here, are clouds of fluorescent spots that are actually very useful if you want to study the internal structure of the chromosomes or their shape or their mingling degree. If you are interested in RNA FISH application more, we can also use the IFISH pipeline to make actually single molecule FISH probes. Here I show you an example of multiple transcripts that we visualized in cryosection from developing human heart and showing that really we have a pipeline for making probes with a lot of possible application. So now before we move to the second part, I'd like to again poll you quickly, asking you what would be the main applications that you can think of what I just showed you. So again, good to see. Oh, that's good. Getting close. Excellent. So we have the winner is high resolution DNA FISH to study chromatin. That's also the main interest in our lab, but also a good portion of you that are interested more in diagnostic application and SM FISH. I want to highlight the fact that what I'm presenting to you today is mainly focused, centered around DNA FISH. However, we are also working towards making our tools, the web tool I'll just show you in a second, applicable also to RNA FISH. So how can we design IFISH probes? So Gabriele in the lab developed this freely available web tool that I invite you to visit. We'll use it in a few seconds. What you can do here are various things. You can browse pre-made probes that we have made and validated. You can download pre-designed oligo databases, and we are expanding the number of databases that will be available. You can also run the software locally on your machine, but most importantly, you can design probes using the tool you will see in a second. But before we go to a real-life example, I'd just like to pinpoint here some of the key features that we keep in mind and that the software actually uses when designing DNA FISH probes. So this is mainly now focusing on DNA FISH. And as you can see here, there are essentially 4 features that we think are important. The upper 3 are relevant when we want to design targets, probes against a single genomic target. The fourth one is more relevant when we want to design what I was mentioning before, the so-called chromosome spotting probes. But for the sake of time today, we focus on the upper three. And so to explain you why we think these properties are important, let me give you and guide you through the example shown here. So let's imagine we want to design a probe consisting of a limited number of oligos, say 96, in this genomic region of interest here that I point with the mouse. So you might have actually a large number of oligos in the database, and so you need to choose how to pick those oligos, and that's essentially what the IFISH for you will help you to do. And so you might want actually to give priority to the size of the probe. So for example, looking at the distance between the first and the last oligos, in this case, you would, for example, discard this region here, and you'd rather choose a region like this one that is shorter. You might also want to actually discard regions where the oligos are not homogeneously spread, as shown here, where you have two clear clusters of oligos, and this might create problem. We can discuss it later. It can create problem by giving split signals. Or you might actually want your probe to be really in the very center as much as possible of your region of interest, and so discard the regions that are more peripheral. So you would end up in picking this region that has a small size, homogeneous spread, and is central. So let's see this in real life by giving an example. So I want to show you how to design a probe, and I chose, for example, the R2 locus. I'm sure that those of you that are familiar with FISH in diagnostic applications know about the importance of this gene in breast cancer. And so what we would typically do is go to a genome browser like the UCSC genome browser and get your region of interest. Of course, you might want to expand then the region a little bit to get a broader view and also design a larger probe. But let's imagine for now to stick really to the R2 gene and look at just its genomic coordinates, which we need then in the probe designer. I need to point out that currently our probe designer only accepts AG19 assembly. So you would first need to convert these coordinates to the AG19 assembly, which you can do here using the liftover tool. You would just copy paste your coordinates here, submit, and you would get your converted coordinates here. Now, for the sake of time, let's simply put some arbitrary coordinates. So more or less 39,700,000 to 728. And let's go to the IFISH probe designer here and see how to design our probe. So first of all, here you can choose whether to design probes for a single target or also spotting probes. In our case, we want to start here with a single locus. We'll name it RB2. Then you have to pick a database of oligos to choose from. At the moment, you can choose from our human database of 40 MERS. You can also use other databases designed with the oligominer tool. Then you would put your chromosome, that would be chromosome 17, and we said, let's try 37 million, from 37,700,000 up to 37,728,000 coordinates. And what you can then specify here is how many oligos do we want per probe. So let's say we want a small probe to have really high resolution visualization. And then here you can output a certain number of candidate probes. I suggest you use the default, otherwise it will take a longer time. And then you have to decide how you want your probes to be ranked and optimized. So for example, you might want to filter out probes that are too large. and then retain only 5 top candidates and rank them, for example, based on their centrality. So how well-centered in the region of interest the probe will be, or alternatively, for example, by homogeneity. So you would then submit your probe, and what you will get here is a summary of your query and a log window that will actually here say, okay, that's interesting, that's good practice. We get an error message. I probably ran some mistake here. Let me see again. I will give you another example. So I wanted to give you also an example here. Let's see. So we can do again chromosome 17, and we can say we can go from 37 million right to 37,500,000. So we will have, there should be here 96 oligomers. And then we can rank like this. Let's try to see if this works. So hopefully this will update here. Yes, and in the meantime, we can copy the query ID so that we can later on go back and see the result. I also wanted to briefly show you one more thing that could go happen here. Imagine you want to now design a much larger probe, so we can again target the similar region but larger. So we can have actually from 37 million to 38 million coordinates, so a 1 megabase region. And here we might actually want to design an entire oligo pool, say 12,000 oligos, 12K oligo pool in this region, really to have a probe that is similar to what you would get from commercial back-based probes. So let's try here to submit, and let's see what we get here. Again, so here we have an error message, and actually this is useful for me to show you what can happen. We asked for 12,000 oligos, but only 9,671 were found in the database. So in this case, you would have to enlarge the region of interest or reduce the number of oligos that you request. Now let's go back to see if it's finished, the first query. So we would go here and paste our query setting, and there you go. So these are our top 5 candidates. Remember, we rank them first, we filter them by size, and then we rank them by homogeneity. And so indeed here, you can see that this probe here is the highest homogeneity, and then lower and lower. So if we pick the top candidate and open it, we'll get some sort of visual checks here, where you can see the location of the oligos that you picked, how the centrality of the probe from here to here compares to the window of genomic region of interest. And this is actually the most important diagnostics here, where we can see the distance between consecutive oligos in the probe. And as you can see here, most of the oligos are around 50-100 nucleotides apart. You can then download the oligo sequences as a FASTA or BET file, and then finally submit your oligos to twist and order the pool. All right, so this was a backup slide. So we would really love to see many people using our tool, but most importantly, giving us feedback, what we can improve, how we can tailor it for community needs and your application. So really, please, we want to hear from you. Just shoot us an email, and we'll be in touch. And lastly, I want to show just a few resources from our lab that I mentioned all along the way. And I'm now open for questions. Thank you.
Thank you, Dr. Crosetto. That was fantastic, and a really enjoyable live demo of the tool as well. It's great to see it being used. So I had a quick question, if I may, to begin with, and then we'll jump over to the questions that are coming in through the Q&A box. So just a reminder, if you do have a question, you can submit it through the Q&A at the bottom. So by contrast to the previous technology, the back-based probes, where you have a single large probe, with the oligo pools, you're obviously tiling across your region. how important is it to achieve homogeneity in your oligo pool? You know, I imagine with a single probe, that's obviously going to be fairly straightforward. But what's the how crucial is that?
I would say that it depends very much on the application. So again, if you are really interested in a well-defined region and you want to cover it as homogeneously as possible. So what we have seen in our experience is that if you have probes where there are these very distinct clusters of oligos apart, and you have high resolution imaging, you will really see the two dots splitting. And that, you know, might be interesting per se if you are interested in chromatin structure and the local organization. If you care more about really quantifying the signals of more, you know, diagnostic application, you really want to avoid this, because you will not know, is it a real signal or is it a false positive? So there, better go with a larger region where you have many oligos, and you will not end up with this problem.
Okay. All right. So I'm going to go with the first question from Pat Heslop Harrison. So the question about the methodology. So why does the probe amplification go through this RNA pol reverse transcription step? Why not simply amplify the DNA? And for most purposes, both strands are both strands okay, or you could use a labeled primer as the last round. So some input there. So any feedback on that?
Yeah, excellent. So first question, why PCR IVD and RT? Well, if you go then to the scheme that I showed you, you can realize that if we were only doing PCR, sorry, we would end up, of course, with double-stranded probe, and we would be facing a similar issue as with back probes, meaning that for DNA FISH application, you will always have the competition between annealing to the genomic target in situ or self-annealing of the two complementary strands. And so in order to make single-stranded DNA, we really need to have this combination of in vitro transcription and then reverse transcription. The second question was whether we can actually incorporate a color directly in the RT step. I'm not sure, maybe Eleni in the backstage can also answer that. In principle, yes, but in order to, so the one very powerful aspect, again, looking at more carefully at the workflow, you will realize that by uncoupling the primary probes from the secondary detection oligos, we actually achieve a lot of flexibility and also reduce a lot the costs, because we only have to order a handful of secondary oligos that are coupled to fluorophores, and then we can use them to detect many different types of probes. If we incorporated the fluorophores directly in the primary probes, this would obviously increase a lot the costs that we face upfront. So I'm not sure if Eleni, you want to add something to that.
I think. Okay, well, I just on a related note then, so you mentioned in your benefit slide that the one of the advantages of the oligo pools based approach is that you get a higher signal to noise ratio with versus the backs. So is that the one of the reasons why as well that you can because of how you're integrating in the labeling?
Yes, exactly. It's again the higher signal to noise is precisely in DNA FISH, right? It's precisely due to the fact that we use single-stranded probes. So just to give you an example, you know, we previously also published a method where we could make probes using PCR only. So that also relates to the previous question. We could generate amplicons, and that worked also very nicely. But still, you will always face the issue that the two strands in your amplicon tend to self-anneal. And so there will be unavoidably a lower signal to noise because of this. Now, with oligo pools and this pipeline, we can make single-stranded probes, and that actually is really solving the problem for DNA fish.
Okay. Next question from James Ravelette. What would be the smallest target that you would feel confident in IFISH's ability to generate high quality resolution? So for example, if you designed oligos against a 10 or 12 kilobase transgene, would this be large enough to yield decent resolution?
Yes, absolutely. So in our experience, I would say we routinely work with targets in the range of 10, 20, 30 kb. We have gone down reliably to detect 3 to 3 kb. I think this is absolutely feasible in our experience in culture cells. So if we talk about tissue, is a different chapter. It's also a lot of issues with autofluorescence. But if I guess you are interested in high resolution imaging of selected candidate regions in culture cells, you can reliably go down to, I would say, 2 kb with our approach.
And in terms of how large you can go, you talked about designing sets across the whole chromosome. So there is a question as well about what's the largest size you could design by IFISH. So.
Right. So we showed already in the paper that, so we compared this chromosome spotting containing a lower or a larger number of probes. So every 5 megabases or versus every 1-2 megabases. We are now actually designing probes every 1 megabase along all the human chromosomes. What we can see is that our ability to estimate the size and volume of chromosome territories varies if we have more probes, but I would say that already with probes every 5 megabases tiled along the chromosome, we can pretty well study properties of the chromosome territories that you would not be easily able to do using standard paint probes. So I would say, again, to start with, if you're interested in chromosome spotting, probes every 5 megabases are a good way to go.
Okay. Well, at least 4 people have asked if you have plans to, for this system for non-human species. Specifically, Daniela's asked about non-human species. Michael's asking about the mouse genome. The mouse genome comes a few times. So, do you have advice for people that want to use this system in non-human models?
That's a great question. I mean, we are obviously aware that, and we want a broad community of people to benefit from this. So that's why we are now running our oligo design pipelines in order to design databases to start with for many species. I mean, all the most important model organisms. So we have almost completed the mouse genome. We want to do zebrafish, C. elegans, and yeast. And so stay tuned. Hopefully we'll soon be able to add our databases to the web tool. And also I would like to add that we are also targeting the transcriptome, starting again human and mouse transcriptomes. So hopefully we'll be able to enrich the tools for the community pretty soon.
Fantastic. And again, so that we also have just for your information, Nicola, someone here working on integrated viral genomes as well. So interested in using it potentially on viral genomes too.
Very interesting. Yeah.
So yes.
Maybe if I can just very briefly add that, of course, if you feel free to contact us if you want to start designing probes against species of interest, where we don't have yet the full database. But you can contact us and we can help you. Happy to help you.
Okay. I think perhaps time for one final question then. So just another technical question then, similar to the previous one about probe densities. So what is the smallest number of oligos that you've tried? Is there a sort of inflection point of efficiency where there's an optimal amount, I think is what we're asking here.
So again, maybe Eleni here knows a little bit more than me because she works in the lab more than me. But I think we have pretty much gone down to 48 oligos with DNA fish. I'm not sure if we tried even smaller region. Eleni, do you remember if you tried a smaller number of oligos?
Yes, we have tried different. I think to be on the safe side, you need to have 48, but there are probes that have worked with 24 also, but well, not always. And also for the SMFIS, I think also you can go much lower than 48. I have gained very good results with 36 probes. But yeah, going to 10 or 20, I think then it starts being shaky on how well you detect the loci.
Thank you, Lenny. All right. Well, on that note, I think we've taken about 10 minutes worth of questions there, and I hope we've covered the key ones. I will try to forward on the remainder of the questions to Dr. Crosetto or to our team, and we can follow up on those. So thank you once again for speaking today, Dr. Crosetto. It's been really fascinating to see the tool in action and hear about how FISH is developed. Just a reminder to our attendees that you're going to be redirected to a brief survey, and we'd be immensely grateful if you could just take 1 minute to give us your feedback. And thank you all for coming, and all have a great day. So bye for now, and see you in the next one.
Thank you. Thank you very much.